So I am not going to do a formal introduction for Dave and Ed, but I will discuss their germline. So, because we know where they are today. So Ed's germline is pretty impressive. Stanford University, WashU, UCSF, and Stanford again. He has been the executive director of the Genome Institute of Singapore and the founding director, and was also president of the Human Genome Organization, or HUGO. David also has a magnificent germline, which includes MIT, Johns Hopkins, the Brigham and Women's Hospital, Dana-Farber, helping develop KIT inhibitors for GIST, developing preclinical models of pancreatic cancer that are KRAS-driven, and Cambridge on the other side of the pond. So welcome back to this side. And this will be coordinated by our two ITSC leaders, and we thank you in advance for all the work you've done in collaboration with us.
Okay, thank you so much, Lee. I also wanted to acknowledge our other co-PI, Larry Baker, who's your past president in SWOG, who is the one really, I think, responsible for bringing this concept to SWOG, bringing Cold Spring Harbor and Jackson Labs in as basic science cancer centers. Now, Ed and I are going to tag team because he has to be on a telephone call in about 7 minutes. So I'm going to just lead off really quickly and go rapidly to some presentation by Ed. You heard from Lee about what we've been doing, which is to bring some of our junior trainees together once a year, either in Bar Harbor or on Long Island, to talk about the science of cancer and new technological approaches we have for cancer, marrying that to some of the clinical conundrums and questions that they bring to the table. This has been very successful, and this year we have 7 new pilot grant applications based upon that meeting we just had 2 weeks ago. And we've awarded so far 14 grants, and you'll hear about results from 2 of them today, one from Ed and one from myself. Lee showed you some pictures of what that looked like. He had perhaps a more spiritual experience at this event than I did, but it was illuminating and enlightening, and we really encourage you to think about who your best trainees are to go next year. It's going to be June 12th through 14th next year, and you'll get that email in the next few months, so you can suggest names. And again, this is to lead to the pilot projects between the trainees and the investigators at Cold Spring Harbor and Jackson Labs. Some of them are very technologically sophisticated, and you'll hear about Ed Liu about what that means in a second. Others are very conceptual, new ways to think about cancer biology, for example, or new approaches for developing diagnostics and therapies. And so I'm going to leave this over to Ed right now, who's really responsible for, I think, coining a term that describes what we've accomplished over these first couple of years, coming up with a very rational approach for combining the best of science and the best of medicine to go after cancer. So Ed.
So part of our ITSC was educational, but in actual fact, it's beyond education. It's actually taking collective experiences, synthesizing them, and having what you might call starter exercises in the pilot studies, as you will actually see. But in the composite capabilities of Cold Spring Harbor and the Jackson Labs, which underscores a subtext that no single institution has it all. Except maybe Arul Chennaiyan's group in Michigan. And the composite expertise that we've had at Cold Spring Harbor and JAX has this virtuous cycle of discovery capabilities, which we called SWOG Discovery Engine, because we become your effectors of discovery. And we rely on you all for the clinical expertise and the clinical nuances that are necessary. So computation, genomics, cell-based screens, these progenitor cell assays, including organoids, then up to xenografts and single-cell genomics, and clinical cancer genomics, which is all capable within the 2 institutions. And when we put it together, we think that working with you, you provide the important clinical questions. We hopefully will provide you the technologic cycling of moving these concepts into practice. Hopefully this will then come back to SWOG in meaningful clinical trials so that everybody improves. What I'm going to talk to you about is how we at JAX, we actually put money to where our beliefs are. And in many of these pilots are shared between the Hope and the Jackson Labs and also the Cold Spring Harbor. So these are the ones where the Jackson Laboratory faculty are involved with. And I'm going to give you an example of one that was started in 2015 with Laurence Baker in Yale and Jeff Chuang at the Jackson Laboratory under genomic analysis of inflammatory breast cancer. And what they had proposed to do is paired-end whole genome sequencing of 20 IBCs and 23 non-IBCs that were extracted from TCGA, which is a wonderful database of genomic information that was matched for age, race, ER, and HER2. When we did this, the initial analysis showed that the two classes had actually a distinctive differences, and these are sort of losses and gains that are prevalent in the inflammatory breast cancers. And more interestingly is actually the point mutations that were found, including some of these that you've seen. One of the most interesting that I flagged was the ESR. That in a de novo IBC, there are that are estrogen receptor mutations, as Arul had mentioned before, suggesting what we see in the clinic, that IBC tend not to respond to hormone alterations. And then, apropos to also what Arul has said, that there are actually not only DNA repair, but actually polymerase type of mutations that are found in IBCs as well. So I'm going to turn it over to Dave for talking about the Cold Spring Harbor.
Okay, thank you, Ed. So you heard from Arul and Dr. Weil this morning about the importance of genomics. We feel that there's another aspect that's very important in trying to think about how you care for your patients, and I call it pharmacomics. So meaning the response of the cancer cells to drugs, empirically, that your patients have. And there's no really easy way of doing that. What Ed and his colleagues have done is perfected the PDX model by developing NSG mice and then various other mice that allow engraftment of human tissue. This is a process that takes some time, but they're the world leaders in it. What we've done is working with Hans Cleaver's worked on cell-based approaches, organoids, for example. These are amazing systems for looking deeply into the tumor. Unfortunately, both of these approaches are way too slow. And the kinetics of both genomics and what I just called pharmacomics are just too slow to help your patient next week. And so we need new ways to think about this problem. I'm going to talk about one last pilot project that we have had between Ann Schott, a leader in SWOG that's well known for all of you, and Mikola Igelblad. Mikola and Ann were thinking about a problem around the role of neutrophils in cancer and whether this could be a promoter of cancer in addition to its response to inflammation that you know is true during infection. And so their project was, again, aimed at a very basic science question, and I will talk about that next. And so the important thing is it actually led to a manuscript that was just accepted by Science Translational Medicine. And so this is an electron micrograph of neutrophils that have degranulated, and the stringy sort of spiderweb material you see there is actually the DNA which has been exuded from the cells upon degranulation, which is an irreversible event. It's been postulated previously that this could promote cancer in some contexts. And so what Mikola is an expert in intravital microscopy, and these are folks who actually can put a window on a tumor in a living organism and watch over hours what happens, what cells go in and out, what happens when you perturb with, for example, a therapy, and what can one learn from such an experiment. And she's learned a lot over the course of her career in this study. So I'm about to play a simple movie. In red, you're looking at a implanted cancer cell line. It's a mouse breast cancer cell line that's known to be metastatic. In red, in green are the neutrophils, which have a gene-targeted fluorescent dye, so you can see them. And if you see blue, this is actually DAPI, so it would be staining DNA. And so you're going to see in real time what happens under the window. And so what you're supposed to appreciate is this cloud of blue, which is emerging from around the cancer cell vicinity as we play this tape. And so this is DNA being exuded by these green cells, by the neutrophils, because they're going away as the blue cloud comes forward. And so she had evidence of this. And now the movie just loves playing. Okay. In this simple mouse model where you implanted cells in a mouse and put your window on a site where the cells had metastasized in the lung. And so she hypothesized that maybe this happened in cancer, and maybe it correlated with how patients did with cancer. You just heard from Ed about a genomic alterations one could find when you compared two different subtypes of cancer. And so just a simple slide on what neutrophils do when they release their DNA. They release it in a formulation which has been described as an extracellular trap, which the concept being that as a neutrophil is activated and degranulates, the NOXs, NADPH oxidases are activated. They make free radical oxygen. And when the cell is activated, the multiple nuclear lobes fuse together. There's actually a lot of DNA alterations which are occurring, including histone modification with citrullination of histones. The chromosomes deconsent, and then the cell spits the DNA out as it's dying. So this whole web comes out of the cell of DNA, which is very viscous, as many of you know. Many proteins are involved in this process, including proteases such as cathepsin G, and these are the steps that occur. When a cell is antagonized by endotoxin, shown above, so bacterial endotoxin would be a terrific promoter of this. There are other ways to make this process occur, as Mikola has discovered. And so the concept that your kids understand better than you, perhaps, or me, is that this is like Spider-Man and his web, which is supposed to be a good thing to go after bacteria. But the question is this a bad thing in cancer? So again, inflammation in cancer, in general, is a promoter of cancer. There's one aspect of the immune response that may help prune away cancer, the acquired immune system with T lymphocytes, et cetera. But there's been this concept out there for a while, and this was a paper by Lorenzo Ferri in JCI talking about this in a model system. And so the question that Mikola and Ann had several years ago is, could you find NETs in breast cancer samples, and would it matter if you could find them? And so they started a project, and they got some samples of human breast cancer supplied by Ann and her coworkers. And so they looked for NETs, and they could look for NETs just histologically by looking for citrulinated histone, which really only happens in the process of degranulation of the neutrophils. And here's some evidence of it right here where you see the green dye. And so then they looked in the samples, and they found that they could describe these NETs much more commonly in triple-negative breast, as well as HER2-positive breast cancers, more so than luminal, which is, of course, was interesting at the time, but again, correlative. And they have a much larger sample set they're looking at now that they have this positive pilot data. But it became the question of what would the mechanism of this be? How would it be that cancer cells could activate neutrophils to degranulate? Cancer cells don't bring endotoxin into the equation. What would be the method by which this would occur? And so Mikola developed a co-culture system where cancer cells were put on top and neutrophils were put on the bottom of a transwell where the cells couldn't get through the membrane. And she could ask experiments in this manner, and over the span of a day, she could see if neutrophils became activated, and she saw that they were activated. And when the neutrophils became activated, they would cause the cancer cells on top to invade into the scaffold. She found that invasion by the cancer cells could be blocked by incubating DNase 1, the enzyme, in with the neutrophils, as shown here compared to there. It then became a question of what was it that the cancer cells were secreting that activated the neutrophils that made them release their DNA. And so she did a variety of assays using the same transwell system that I just described. And by doing so, she, of course, consulted the literature. There was a nice paper by Dave Skadden talking about how neutrophils could aggregate with platelets to actually promote microangiopathy that you find sometimes in cancer patients. And she looked through a variety of methods for the secreted factor from cancer cells, and she found that it was actually granulocyte colony stimulating factor, because she could do additive or subtractive experiments, identify G-CSF, inhibit it with an antibody, or add back G-CSF to actually promote this. And so you could actually make the signal go away with the antibody. You could create the signal by putting in the ligand. So this was obviously interesting. This would be something that would cause chemotaxis of granulocytes as well as, in this case, activation. And so the question became, now that you know this, could you actually interrupt using model systems? And so this was difficult, a challenging approach for Mikola, but by actually working with Anne and talking to many people who are interested in this approach, she was lucky enough to interact with a bioengineer at Harvard who talked to her about various approaches she could use to interrupt this. She was able to take DNase 1, the same enzyme we give CF patients when they're having flares by inhalant, and actually use it as a conjugate in a nanoparticle, injecting it systemically into a mouse model. And she could show that she could knock down the NET formation in these animals. And so this is an experiment comparing, again, microparticles, nanoformulations of DNase 1, in this case in green, compared to just injecting DNase 1 in free fashion in purple, showing that you could actually really reduce that the DNA levels much more readily by injecting it in the preparation. And when doing so, if you compare green to red, you decrease the number of NETs in this model system rather dramatically. And of course, the very exciting information is shown here. These are just H&Es showing the mice that received the control treatment of the nanoparticles compared to those that received the DNase 1 coded nanoparticles, showing you the huge difference in metastatic burden in the mouse lung after they were set up in a metastatic model. And so this was, again, a preclinical suggestive observation. I guess in a rules list, it would be tier 4. Publication in a science journal, promising, but something that we could think about actually in getting to the next level up. And the conclusions from this, again, very pilot-based experiment was no fancy drugs, just a biological question, using model systems and the intuition and know-how of basic scientists working with a very excellent clinical oncologist who wants better treatments for our patients. That these neutrophils, which we all need, and we all we worry about when the levels get too low when we give cytotoxic drugs, it may be a double-edged sword with them. And if they get activated in the wrong way, they may be promoting the cancer process. Of course, there's this idea when our patients get neutropenic, we give them growth factor support. We do this because we don't want them to die of sepsis, obviously, but could we be promoting this process of cancer progression? There's an observation, I don't know how controlled it is in the literature, that when you get an infection after cancer, you have an increased risk of metastatic recurrence. This, of course, is antithetical to the Colles toxin observation 100 years ago, but this is more in the modern era. And why is that? Why is there a bigger increased risk of metastatic recurrence? It would be great if you could ask questions in those patients if the neutrophils in their nets had something to do with it. And so this, again, just leads to another option, another avenue to think about in this disease. And this is Mikola and her coworkers with the funding, again, coming from this very discrete pilot award, leading to something that possibly will be bigger and relevant to our patients. And I just wanted to end with a real thanks to the Hope Foundation that Ann Schott and Lee Ellis are big contributors towards, and the officers for, help started by Larry Baker. And also to Charles Blanke, the current president of SWOG, who have all been very supportive of this idea of bringing basic science cancer centers into a very large clinical cooperative group. We hope this is synergistic, and we thank you for your financial support. We look forward to more exciting projects. Thank you.